Thank you so much, and thank you for the Associazione for organizing this, for bringing all the clinicians, researchers together. I'm an add-on, and I'm going to try to divert you from COL4A1, maybe bring you back at the very end, so be prepared. We're going to be talking about other diseases. And I thought about sharing my experience on doing gene therapy-based treatments with families, so patient-centered, patient-driven drug development. I'm going to need some help with some videos in the back, which he's standing, if he can activate. These are my disclosures. I do advise in a lot of different companies. We primarily are funded by NIH, and we also have other people that are trying to get us to do the research. And of course, we wouldn't be where we are if we didn't have philanthropy from the foundations themselves. That's how we got the first preliminary data that we put for NIH funding. That's what I use this slide for. We are in Boston. This is Mass General. It happens to be a hospital that has from before you're born, basically, care until you are an elder person. So this allows us to encompass and grab the entire lifespan when we are working on genetic disorders. This is the vascular gene therapy team that I direct right now, and it's a combination of 5 different institutions. This is really hard work. It takes a lot of effort and a lot of expertise, so you can see what they are experts on, and this is just to treat one disease of 55 kids in the world. So is it feasible? Yes. Can we repeat this and do it better? I'm convinced about it, but we had to get there. So bear with me on this. And this is our vision. We really are looking into treating one patient at a time, but with all in mind. And I'm gonna try to walk you through what are we enabling when we are doing treatments in patients on the technology side that could be applied to many, many other diseases, not even genetic disorders. And you heard Elizabeth talking about how COL4A1 is also involved in common non-genetic vascular disease. These are the objectives, and I'm going to walk you through 2 diseases that you may or may not have heard, and the one thing in common that I always use to conceptualize, so I started as a stroke critical care neurologist. I encompass pediatric and adult care, both, and I'm part of the very early teams of leukodystrophy gene therapy with Dr. Eichler, Florian Eichler. And COL4A1 is still listed as one of the leukodystrophies, and I want to put that pin until the end of the talk to say they do have a global clinical trial network. So both in Europe and in the US. So we could potentially utilize that infrastructure to really get all the parts that we're missing to get to treatments for the different phenotypes of COL4A1. And now I'm gonna say it's easy. COL4A1, you guys heard the phenotypic descriptions were outstanding this morning. You heard about the genetics that underpin this. And I literally learning from Doug on what do we need to do if we're gonna try to improve this disease, because I am not the expert, and that's also one lesson I have. You don't have to be the expert in everything. You have to bring the experts together. So a little bit of an introduction, and this is, I always use this quiz because in neurology, people use imaging to diagnose leukodystrophies or white matter disease, and you can spend quite some time on this and try to guess which disease is underpinning this. And in reality, this is what we do in the clinic, except that you have a center like in France where you can send the patient for an evaluation and a genetic evaluation, but usually we bring a bunch of people, we call everybody we can think of, knows about a possibility of that patient having the disease. And here is a list of those diseases, and that's why I pin back into small vessel disease, which is a common denominator for COL4A1 phenotypes in the eye, in the brain, in the posterior circulation, in the anterior circulation. And COL4A1 has some features that we can leverage even in the biomarker studies. And why are vessels so important in the brain? Every single stem cell in the vessel depends on normal neurovascular and the perivascular niche working. So I have a lot of respect for little capillaries. This is just a list of those genes that, in a very summarized way, in a vessel wall, this is the vessel, that's the wall, you can see endothelium, smooth muscle, and the extracellular matrix. And the different diseases and disorders, it kind of feels like every single one has anywhere from a couple of handful of patients to hundreds of patients. So there are some not so rare. And that's, I think, something that you also think about it. And COL4A1 is one of the not so rare compared to others. So when I conceptualize how do gene and genetic disorders try to alter vascular disease, I'm thinking about the brain phenotype. They're basically these very basic phenotypes: white matter injury, ischemic stroke, hemorrhagic stroke, and of course vascular dementia. I just summarized it there, but disruption of blood-brain barrier function and microvascular or neurovascular coupling does not allow a network of neurons to work. So that will come with processing impairments and cognitive inhibition. And this is the schematic of those. So gene-targeted approaches. For those of you that haven't thought a lot about gene therapy, if we summarize it for vascular disease, I think we're going to be talking always about systemic delivery, ideal. We can still go to the subarachnoid space and try to target vessels there with intrathecal. We are going to have to try to go, as Doug was talking about, working to modify protein function with the chaperones. We can modify the RNA, which is a step prior to that, with modifying the expression of an RNA, either microRNA, siRNA, or ASOs. And we can directly target a gene. When we say gene therapy in general, we're talking about this one. Either you put back the gene that is missing, not the case for most COL4A1s, and gonna be not easy to do that. And gene editing, which is what Doug was referring to, and we're working together to develop. And in general, in gene therapy, you can do gene therapy inside the body. We call it in vivo, or you can take stuff from the body, change it in the dish, and put it back into the patient. That we call it ex vivo. So I'm going to give you an example of ex vivo gene therapy. I have been involved from the very first day that these patients were being treated. And then I'm going to tell you a preclinical that we're in front of the FDA for a genetic vasculopathy that is less common than COL4A1, which I'm hoping will enable. So how do we get gene therapy to the cell? So how do you change the gene? How do you add the gene? These are our two main vectors at the time that we are talking about. There is also HSV vectors, adenovirus vectors, but these AAV has more than 2,000 patients treated already in gene therapy. So we know what AAV looks like, so in humans. And it's a small virus, doesn't infect you, doesn't give you any cold like I have right now, but it will be able to cargo, to take a payload, but a very small one. So you have to also think about what you need to bring into the cell. Lentivirus is a lot bigger, but these guys will put the material into your DNA, genomic DNA. So they have other risks, and I'll tell you a little bit about that. We have to think about where you need to deliver the therapy. This is very important. You have to make your choices. And in COL4A1, with the phenotypic complexity, we're going to have to pick what are we treating first. And it might be the eye. And that will be an excellent thing for the patients. The corneal opacities continue to be disabling. Of course, retinal hemorrhages are very disabling and can cause blindness. So I'm going to walk you very briefly through the journal from natural history, which is, I think, what this conference and the round table that started with the Associazione like a couple months ago, like in November. This is the very first step, and you can't skip it. If you don't know where, when, who to treat, you're not going to have, no authority are going to let you treat a human, okay? So you're going to have to show understanding of the disease and why you're doing what you're doing when you say that you're doing it to the organ that you target. So this is an FDA-approved gene therapy. We start with the disease that affects, is the X-linked, it's adrenal leukodystrophy, it's the most common leukodystrophy. And you can see there's different phenotypes, not as complex as COL4A1. Adrenal insufficiency, a cerebral, horrendous demyelinating disorder that causes like extensive white matter degeneration in a record amount of time. In 1 to 2 years, you lose your ability to communicate. So patients are severely disabled once this disease starts. It has 2 peaks, kids and adults. And then if you are a patient with this mutation and you're a man, you will develop AMN. If you're a woman, you will develop AMN, a myeloneuropathy, a problem with the nerves that puts you in a wheelchair and has a lot of other disabilities involved with it. So if we can play the video. This is one of our patients. For those of you, this is a 6-year-old, 7-year-old here that is in his karate class. He did not know the disease was in his family. He didn't know he had a mutation on the ABCD1 gene. And if we can play the next video, you can see what the disease looks like. He started having a little bit of behavioral changes, running into walls, his peripheral visual field was affected, And this is unfortunately only 12 months later after this karate practice. So this is the degeneration of white matter. This disease gives us a lot of a window to do things because we don't have anything to offer them. And we did have one learning lesson from compassionate use. And again, a lot of what we learn is from the human experiments, from treating kids and families. And one of those was done here in Paris by Patrick Abro at the time that Hugo Moser was treated in Kennedy Krieger, and they did a bone marrow transplant. They said, well, this looks like inflammation in the brain. What if we change the immune system that lives in the bone marrow? Hopefully, we get normal immune cells with a gene, with ABCD1, that can go to the brain. This is all hypothesis. And change and modify the immune system in the brain, and maybe that stops the demyelination. This was treated, Kennedy failed, the kid progressed, horrendous disease. France succeeded in rescuing and halting the disease, becomes the standard of care for these kids, continues to be today standard of care. Transplant the kid as soon as you diagnose them with a cerebral lesion. And because we knew transplant could rescue them, when you leave them untreated, you will lose almost all neurological function. And this here took 25 years of developing scales to capture the disease severity. So what we are doing today is critical. We have to be able to think about what is the rate of progression, what progresses, how are we going to capture it, and most likely it's not going to be in a scale that we have somewhere else, that we just grab from the drawer. This was the goal. We give bone marrow cells, we put cells that go to the bone marrow, replace those cells, and they will make these beautiful microglial cells. This is the observation from Dr. Eichler in the brain of these patients. And I hopped on something when I was a resident. I wasn't even graduated from my training. And we started with Bluebird Bio, was a biotech that got founded with the gene therapy, again, that an academic guy did, Patrick Avro, in France, did the first ex vivo gene therapy. And it was his work that got acquired by this little tiny biotech and went to Florian and say, hey, can we try to do this again? And in a larger scale. So we took it on. This is Christine Duncan, and she's the hematologist that is absolutely needed because the whole work is doing a bone marrow transplant. So basically, more than 2/3 of the boys don't have a donor. So we were seeing kids that we know what to do, but we don't have any treatment to offer because there is no donor for bone marrow. So these are their own cells. We use a lentivirus. We put the ABCD1 gene back. We give them horrendous chemotherapy. Okay, this is not benign. They can die, they can have all kinds of things. And then we hopefully see that they're engrafted and they go to the brain. The FDA asked us to treat one kid at a time, and until that kid engrafted, we couldn't treat the next one, and that was for a whole year. Meanwhile, we were seeing the kids of other and siblings deteriorating with the disease. So this is not easy. Nobody sleeps when you start clinical trials in gene therapy. Thanks goodness, this is conditionally approved, and it provides another option for the patients. Again, standard of care is bone marrow transplant. If you have a donor, go for it. Don't go for gene therapy, and I'm telling you this, having been a very big part of this, because we find out later on that there are side effects, and side effects are myelodysplastic syndrome, and it's gonna be around 1/3 of the patients having this complication. So you only do it when you don't have anything else to offer, and the parents are always, and the patient will be the ones that have to make the decision, do I wanna take that risk? And it has a treatment, no? So MSD, you can do another bone marrow transplant, suboptimal, and you just fix them and they don't get a leukemia. And we have every single patient in monitoring for 15 years after we dose them. So not easy trials to make. No gene therapy is an easy trial. This is what happened to the lesions in the brain. If you don't treat the kid like this is what it would look like. And if you treat it, you can see how the treatment completely arrested the disease in a quite a rapid manner. So we were very happy to see these boys. This was not possible without 5 different patient foundation advocacy groups. They not only funded the research that was basic, including Patrick, which ALD Association by 2 sisters that got funded by them, but they also have, their own family has experience of having a kid affected with the disease that did not get the treatment, was never diagnosed, and a brother usually that gets the ability to be diagnosed. We have newborn screening now, most getting established in Europe, mandatory in the US. We still have half of the states enable. And these are the first 3 boys that got the gene therapy. These are their initials, and for a neurologist, and EDL is an activity of daily living, and ALD are the initials of the disease. So they had this idea, the kids after the transplant. You can see them that the chemotherapy has taken their hair for some time. And the first kiddo in that picture is the one that get MSD 7 years later. So again, victory and not so, okay? So that's always things that happen when you start intervening with biology. And Doug was pointing to also this critical understanding of, are we gonna make something really worse? We get a lot of collagen out, and that's what we do with our treatments. Are we helping them? So these are just MRIs cycling, and that's the lesion, and you can see how it progresses. And we had an observation during the bone marrow research that was this microvasculature, the small vessel disease, is a lot more expansive than that. That led to 3 clinical trials to different therapeutics. We know these changes precede the appearance of a tiny little lesion in the white matter by more than 12 months until they get contrast. So we have an earlier biomarker of disease. Can we do preventive treatment? And a little bit with both in mind, trying to be preventive and rescue. We know you lose your gene. This is very applicable to COL4A1. We know we have a genetic defect, but there is a ton of other factors, second events, that make the vasculature be dysfunctional. And we have now, this is about to be published, the preliminary data for a leriglitazone, which is a PPR gamma agonist. And again, most of these patients that I'm talking about, and I'm from Argentina, I live in Boston, and a lot of the patients from Latin America seek consultation with us. They are not going to get gene therapy. It's only approved in the US. Europe didn't authorize it, even though it got approved to be commercialized in Europe. So the majority of patients do not access gene therapy. And a bone marrow transplant in Latin America is 8 months to 12 months to get transplanted. The disease doesn't give you that time. So my aim, to get a drug that slows them down so we can get to transplant. And apparently we're doing that, and these are the rates of progression of natural history. Again, natural history, biomarker imaging. And this is the rates of progression of the disease. An international patient academy industry collaboration, again, highlighting the extreme need of working together. And this is the one that I wanted to share as the last example. And this is the systemic gene therapy for a genetic vascular disorder. And the idea is, in this case, we want to do targeted gene correction. And there are many different ways that you can do that. And some of them use enzymes. These enzymes came from bacteria. They're called CRISPR-Cas9. And there are many iterations of those enzymes. And you can cut DNA and try to use a donor or use the same repair mechanisms the cell has, or you can do something much more elegant without cutting. You get in between the 2 strands of DNA, and you're able to use another enzyme attached to it with deaminases, for example. So it changes 1 letter without cutting DNA. So think about the off-targets and stuff. This is a lot more manageable in terms of that. Not only that, for COL4A1, very importantly, you can use these enzymes to really find the place and try to upregulate and downregulate. I'm humbled by what we need to learn about up and down regulation of COL4A1 and 2 to know if we have a window there. Okay, there is no treatment if we cannot get to these cells. And timing is critical. We know, as Doug was mentioning, 100 years of collagen in your body. That's the lifespan of the collagen. So we're probably going to be talking about treatments that are very early in childhood as a first treatment. But you need to get to these vessel cells. And in this case, smooth muscle was very important, endothelium as well. And how do you get there? This is AAVs. And you can see brain pericytes, beautifully transduced. We developed this one from an AAV9 vector. We were really lucky to get a lot of a smooth muscle cell in large arteries as well. And that gives us a little bit of hope that we could do a proof of concept. This is unpublished data, and this is the vector in human arteries. So we are a hospital, and we have arteries coming from amputations, and we are trialing all the vectors in those, and you can see how beautifully this guy transduced versus AAV9, which is the most commonly used. And things take a process. Things don't happen from one day to another one. I just want to recall that COL4A1 was first ever associated with porencephaly and prenatal hemorrhage in 2005. We are in the explosion phase. There's 350 clinical trials using AAV9 right now. There are 1,400 clinical trials using MRIs, siRNA, and ASOs at the moment. So there is a lot going on, and we are going to learn together with other people with other diseases. And one dream, we will have not a vector that is a virus, we will have a nanoparticle that gives us a lot of more flexibilities on how to deliver the therapeutics, including in small molecules that you can target to specific cells. So this is a disease that I wanted to share very briefly what we've done. And They can be diagnosed like in COL4A1 because of an eye phenotype. Instead of having colobomas and cataracts, congenital cataracts, they have fixed dilated pupils. They look like they're herniating. And that plus a PDA is the diagnosis of ACTA2, and it's almost no differential. So if we can get these kids early on, which we're doing, we can hopefully have windows for treatment, at least preventive treatments for a stroke. They do get narrowing of the vessels, so it's not mostly hemorrhage, it's mostly ischemic strokes that they're suffering. It is a very multisystemic disease of the smooth muscle. So everything that has a smooth muscle is dysfunctional. This is one of my patients that I'm gonna skip. This is Rini, and you can see her here at almost 8.5 years of age, and she's singing Frozen. And her vasculature, we don't have her MRI, but this is from another patient. At 10 days, they're still not critically stenosed, but as they age, this is a 7-year-old MRI, they do develop this terminal ICA stenosis while the petrous portion dilates. That gives us the biomarker for treatment, by the way. This is what we're putting in front of the FDA. And how many patients did you have? 27. And how many did you find this? In all of them. So it doesn't have to be hundreds of patients to understand what you need to know that justifies the risk. And this is unfortunately her after multiple strokes. This is another lesson. This mom and a group of parents started to found a foundation. Before Rini, 33 patients in the history of the disease. This is even newer. The gene was first identified in 2010. So dog, you were 5 years ahead on COL4A1. And since Rini has been diagnosed, we have 24 patients in 4 years. So you can see rare is rare while we are not knowing and we're not looking. And we are now accessing at a much higher speed whole exome sequencing. I think the numbers in COL4A1 are gonna be very high. According to all the things that we're seeing, 20% of prenatal hemorrhages are COL4A1 related. It's mind-blowing. So this is what I always say, developing therapies for them, with them, and the engine to everything. I don't sleep, but I don't sleep because I have these guys. In fact, there is one in Insern right now in the ICU, and there are 2 in Boston, there's 1 in Texas, in the critical care unit trying to save them from all the complications that they're having. The biomarker that enabled the possibility of a trial, we knew that this ratio matters. The stroke happens only if you're really stenosed in your carotid. That is not found in controls. That's another thing. We need just a biomarker that is very unique to COL4A1. And perfusion imaging may be the answer, by the way. So this is an schematic of the whole story, and I cannot show you a patient treated. We're trying to get the first patient treated in 2025. We are on track with that. A lot of work precedes our work with Diana Milowicz and the natural history. We're hoping for a prevented therapeutics in the first few years of life to not let the brain vasculature narrow. We might end up with a rescue for the aorta. These kids have aortic dissections around 14 years of age, and they also die before 20. So the kids can present with Prumbelli. You can see the cytoskeleton. This is ACTA in green, and the fibrillary actin in red that is supposed to be made. This is a control, human, and a control, a mutant, Arginine-179. This is a mouse, and the mouse has the exact same phenotype, and we got really lucky. The mouse has the entire phenotype. It has the narrowing of the vasculature in the brain. It has the rectification of those vessels with the hypoplasia. It has the entire systemic thing I'm not going to tell you, but gastroparesis, blah, 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 fixed dilated pupils. We could not believe and get luckier. I'm going to make a pause. Gene therapy for ALD was done without mouse model. There is no mouse model of ALD, cerebral, and we could have generated up to now, 40 years since the gene has been described. So not everything is linear. It's a very different combination of things that allow you to get to a treatment. So we had to generate a cell line, and I'll tell you, we've done this now with dog to be able to model how we can change it back to the residue that needs to be. This takes a lot of combinations. There are probably 1 or 2 years of optimizing the editing strategy. You finally get something that you like. You take it to the mouse. Sometimes it's too big to put in one single AAV. I told you they are small. So these enzymes are large, and sometimes we had to split it. We did that, treated the mouse very early. And if you can click on the videos in the cages of the mouse, you can see an ACTA2 and a wild type. These are littermates. They can have very little mobility. And if you click on the other video, you guys can see you cannot differentiate them. We have to go to the tail and genotype them to know that this is a treated animal. We were able to rescue a significant amount of survival, the activity in behavioral phenotype, and this was unexpected. We thought the aorta was a completely impossible target, but apparently we're doing what we need to do, and both treated with AAV9 and the new vector is showing that the aortas are not dilating. And this effect is now 9 months long, and the aortas are still normal size. So in the brain, did we get the brain? Can we get something like a CRISPR-Cas9 enzyme inside the vasculature of the brain? The answer is yes. This is a control. This is a mutant artery. You can see there is no contractility. There is no wrinkle. And then this is after it's treated. And there is no need to do grafting or anything. They're very, very rescue. So smooth muscle cell can be targeted in vivo, can get editing, and it can rescue function. Did we change the white matter disease? So I didn't tell you, but this mouse has horrendous loss of myelin. And you can see it here, and these are the treatments, and the answer is yes. We are doing a perinatal treatment. So this is very early on. Blood-brain barrier is also rescue. This is GFAP as a marker of disrupted blood-brain barrier, and these are the 2 treated animals on GFAP in the brain. And I can show you another 20 stains, but that's not the point. The point is vessels are a key component of neurodegeneration. And I think for COL4A1, this has been proved across every mutation and every phenotype. So if we can treat microvessels, we will have a significant impact, I think, in the disease. There are videos here, and this is the gut and every organ, but the gut gets impacted, they don't move it. If they're mutant, this, you can see there is no impaction, and these mice, that was another bonus. We weren't expecting to hit the gut. And that happens when you're doing it. We don't know what is gonna happen in the human. Do we edit? We edit up to 50% in the liver, 30% in the heart, or 20% in the heart, and the brain vasculature is single digits. You don't need that much. And we're doing single RNA-seq to see what is real, the number here, because we have endothelium and a lot of other cells that we're not targeting. But that's, in the aorta, I was even more surprised, like 2%? 2% of the smooth muscle cells in the aorta you need to change. Nothing is simple. We gotta go from little mouse to baby, and we're trying to create a sheep model, a pig model, any model that could give us how do we scale up and do it safely. The dosing is one of the main things that we're trying to estimate. All of this, the vector, the enzyme, the dosing, the large animal, transfers from ACTA2 to COL4A1 to the next disease, because we're trying to target the same cells pretty much in the same time of development. And back to COL4A1, a couple of slides. You need to recreate this cycle over and over and over for every disease you decide to try to do something for the patients. And I'm going to put, there is not just gene therapy, there's a lot of things that we do when we engage with the families that improve care, and one of them is the multidisciplinary clinics. And Rachel Bazar here in the audience is trying to start this. The Europeans, you guys are way ahead of us. You have centralized rare disease, everything, by repositories, genetic testing for most countries. It's really humbling to see the advancement, and that's why most of the literature on what these diseases look like is coming from the European side. The US hasn't put their act together on how they're gonna get people diagnosed with genetic testing, and as sad as it is. We have for COL4A1 to understand specifically what we're missing and work towards getting those. Do we miss the biomarker? Do we have natural history? Do we know what outcome measure we're gonna be targeting? And that we do with the patients. There is no way, this is rare diseases. It's not that you're gonna go to a hospital emergency room and you're gonna find them. You're gonna find them because you work with the Associazione for the Famiglie. And this is one of my patients, again, highlighting the delays in diagnosis and the classic COL4A1. I don't want to repeat, the phenotype is very wide. I think this phenotype is under-recognized because a lot of patients, mostly in pediatrics, also don't get MRAs of the brain. And I'm seeing the fusiform dilations the more I look. So I think what you don't look, you never found. We talk about this, the small vessel disease. I think it's going to be the target. You can see all the implications of having a small vessel disorder. Which is not just the MRI white signal in T2. And of course, if we can restore vessel wall integrity, we're going to be able to prevent some of the most tragic parts of the disease, which is the hemorrhages. So what are we doing with dog? Dog has a collection of mice that we can try, and we have to think really careful because each of these iterations is very labor intensive, to the point that we did some of this, and then we decided to go for the most common mutation you can find in humans and try to create another mouse, even though he has many, many mice that have genetic mutations. So we humanize a mouse. And we're not yet with a mouse back, so we don't have in vivo, and this is all gonna stay tuned. But basically, we created a cell line, human cell line, and we are doing very different iterations for different mutations to see what exactly we can do for each mutation. And each mutation is a lot of work. I'm very positive the FDA is putting a white paper on how we're gonna change the guides and use the same enzymes. And if you treated one patient, they're going to let you treat the next one, as far as the endpoints are similar to the ones between one patient to the other. So a lot happening. You saw the work that Doug is capable of doing of human cells and how we can see if that retention of collagen, it starts to be secreted. So there is modeling, and this is just a pin there for mostly the US families and clinicians. There is something called Global Leukodystrophy Initiative, GLIA. Adeline de Wever at CHOP is the leader of this. And COL4A1 is still listed as a vascular leukodystrophy. So we have to convince them to add us on. And you can see in other leukodystrophies how they are doing amazing work on characterizing the disease, and they're trying to get them ready for clinical trials. So this network is trying to do things for rare diseases where there are no resources, but by adding them to a network that has the resources. NIH is funding this network, so I think we should push for getting COL4A1 added there. Frequent enough, guys. They rejected ACTA2. I tried them. They said, no, 55 patients, too little. So I think the most important thing in this slide is not one size fits all, and I will leave it with that. And the only way to start, continue, and get to any line of success is if we work with the families all the time. So with that, I'm gonna skip. We also do all this pediatric educational series if you need for rehab. And I just wanted to put a shout out to my lab. Dr. Lindsay is a cardiologist that is an expert on the smooth muscle, and a much larger collaboration with very different collaborators across different universities, and a little philanthropic guy that formed a company to give research agreements to get us accelerated in whatever we're doing. And that's thanks to Charles Roswell, which is this guy here. And with that, I think we don't have time for anything. So lunch? Thank you so much.